Good morning, everyone. I'm Lori Burrows. I'm the Associate Director of the IIDR. Welcome to April's edition of IIDR Rounds. I'm super excited about today's talks, which are about my most favorite topic in the whole world, which is multidrug-resistant Pseudomonas. So our speakers today are Dr. Cheryl Main, who is a medical microbiologist and infectious disease physician who has been working in the Hamilton Regional Lab Medicine Program since 2004. Her focus in the lab is on bacteriology, quality improvement, change management, and lab utilization. Her clinical responsibilities include inpatient and outpatient clinical infectious disease consultations at the Hamilton General, and Dr. Main serves McMaster as a professor of pathology and molecular medicine. Today, she's going to be speaking to us about the clinical challenges posed by Pseudomonas aeruginosa. Our second speaker today is Dr. Brent Weber, who is a postdoc fellow in the lab of Dr. Eric Brown. And I want to thank Brent for getting up super early today because he's actually coming to us from Alberta, where he is working temporarily. His research focuses on identifying new antibacterial compounds with activity against gram-negative pathogens. And today he's going to be discussing the antibacterial of a synthetic siderophore monobactam conjugate. So Cheryl, take it away.
All right, good morning, everyone. So Lori and I have in common that Pseudomonas is our favorite drug or bug. And I have to say that it is the one that I get most excited about in the lab when I'm working as a microbiologist, but when I'm a clinician, it's the one that gives me the most fear. So I'm gonna, I have no affiliations or disclosures. I'm gonna try and share some of the clinical challenges that I've faced when dealing with multidrug-resistant Pseudomonas and identify some of the gaps in current therapies. And I'll also share a little bit of our lab experience with this organism. So I'm going to start with a very complicated patient who I started seeing several years ago. I didn't start seeing him when he first presented with issues. So at the age of 15, which was 1985, unfortunately, he developed leg weakness and was found to have a myopapillary epidymoma tumor, which is a really aggressive spinal tumor, which required multiple surgeries to debride it. He had resection of the tumor all the way from T6 down to his sacral area. So you can see that's a huge distance. And in order to stabilize his spine after the surgery, they had to put in hardware. So I've shown you a picture, an X-ray here of what similar hardware looks like. It's a lot of metal rods and screws that they have to put in. Unfortunately, despite that, he did become paraplegic, even with all of the measures to avoid it. He had a long course of recurrent surgeries, radiation therapy, and as a result, he had fairly poor wound healing. He had a few sinus tracts along the incision area, which isn't uncommon when we do radiation therapy on these patients. He did require a urinary catheter because of his paraplegia and did become colonized with multidrug-resistant Pseudomonas, but was otherwise doing well until about 20 years later when he developed an abscess in his spine due to Staphylococcus aureus. This was treated appropriately. He had surgery, they debrided the abscess, they did exchange some of the hardware. One of the challenges when you have these infections where hardware is involved is that you get biofilm on the hardware, and that's very difficult to eradicate. So they did change some pieces. The additional issue we face with these patients is sometimes the hardware becomes unstable when there's infection. He was treated for his Staphylococcus aureus with cloxacillin, which is a fairly routine practice, but developed renal failure and was found to have an interstitial nephritis, which is an unusual complication from cloxacillin. All in all, though, they managed to clear his infection with 6 weeks of therapy. He was left with some more open sinuses along the wound, which they were managing with dressings, and he did well until 5 years later when he had another Staphylococcus aureus bacteremia. He was again treated, and at that point, the decision was, with all of these sinuses and the hardware in his back, we're going to put him on suppressive therapy. So we do that when we can't fix the problem in ID. And so doxycycline once a day is what he was to be on. At that point, my colleague who was managing this patient left Hamilton and said, would you take over this lovely patient? And he came to me. And I was seeing him about once a year to ensure that I reordered his doxycycline, that he was tolerating it, and that all was well. Fast forward to 2016. So I saw the patient early in this year. He was doing well, told him to stay on the doxycycline, but unbeknownst to me, he decided to stop it 2 months later. He told me it was, in hindsight, he said it was bothering his stomach, but he didn't call me, he just stopped it. And within 3 weeks, he had increased drainage from his wound. I received a call from his father who said he's having fevers and the wound looks red. And I said, get to the hospital. And at that point, he was found to be bacteremic with Staphylococcus aureus and Klebsiella pneumonia. Again, he was treated. He was discharged home on ertapenem and was stable from that perspective, but he came to see me in follow-up and he had a large, hard lump in his back, which I suspected was hardware becoming unstable. So I mentioned he has lots of screws and rods in his back, and one of the rods had come loose and was poking out and would have eroded through the skin. So I arranged for him to see his spine surgeon, and he was scheduled for repeat surgery. And that's when things got exciting. In the surgery, they sent off a bunch of tissues. I think they sent off 6, and one of them grew 1 colony of a multidrug-resistant Pseudomonas with this pattern. So at this point, the organism is susceptible to piperacillin tazobactam and the aminoglycosides and colistin, but resistant to ciprofloxacin and meropenem, which is interesting because he had not been on meropenem at any point, to my knowledge, and intermediate to ceftazidime. So with this, the decision was to treat him with piperacillin tazobactam and vancomycin for the Staph aureus, and he was also given rifampin, and he took all of those drugs for 8 weeks. His wound healed up. The hardware was now stable, and he came to see me before they removed the sutures, and I said everything looked good, and so I said, go ahead, remove the sutures. And then that's when things got really interesting. And if anybody's eating breakfast online right now, I'd recommend you might want to divert your eyes from the next picture. So I received a call from his father who said his wound has opened up after the sutures came out, about 48 hours later. So I sent him back to the hospital, and his wound looks similar to this. So it was big. It was 28 centimeters by 10 centimeters with exposed hardware. This picture actually was taken sort of into therapy, so that it was much more red and had a lot of purulent drainage. You can see there's a lot of exposed hardware that is covered in biofilm. So at this point, obviously, there was a lot of concern about this. He was started on a vac. We put a vac dressing on it. And for those of you who don't know what a vac dressing is, it looks similar to this. So it is a dressing that has a sponge that goes into the wound bed, and it basically acts as a vacuum and sucks the fluid out. So it's helpful to remove fluid that's accumulating in the wound, and it also helps the wound heal from the base up. He was also put back on piperacillin tazobactam initially, a high dose with an extended infusion to try and really kill that Pseudomonas. He had a little bit of tobramycin and vancomycin. Wound swabs from that admission subsequently grew this bug. So you can see the Pseudomonas has become more resistant. So now it's resistant to the aminoglycosides. We can compare it to the previous, where it was previously susceptible to piperacillin tazobactam. Now it's intermediate. It was previously susceptible to the aminoglycosides. It is still intermediate to ceftazidime. We did arrange to get additional testing done, and it was susceptible to a brand new drug at the time, which was ceftolozane tazobactam. It was a drug that wasn't fully licensed in Canada at that point. So he was tried on a number of things. He had this vac dressing on. He was on ceftazidime. There was a little bit of healing of the wound, but not completely gone, and it made him quite nauseous. At one point, amikacin was added, but his creatinine went up by 3-fold, and he had some ringing in his ears, so he wasn't tolerating that. We were able to miraculously get ceftolozane tazobactam, and this resulted in increased creatinine. He was also nauseous with it, and his potassium went quite high to the point where we had to hold some doses. We did a trial of colistin, and this actually not only caused his creatinine to go up, but gave him significant arm weakness. So we measured grip strength, and his grip strength dropped by 30%, which is a huge side effect in somebody who's paraplegic and relies on his arms. And despite all of these therapies, every time we swabbed this wound, we grew multi-resistant Pseudomonas. And at this point, his wound is looking a bit better, but there's still an awful lot of exposed hardware. And at this point, the patient said to me, "You're killing me with these treatments." So he had so many toxicities from all the antibiotics. He said, "I don't want any more, and I don't care if I die." And his dad came to me and said, "Can't you do something different?" So that I started scratching my head, and I went to the literature, and I found an interesting article, which is here. And this was a study of patients with chronic MRSA infection of prosthetic joints. So they've been infected for more than 3 months, where they surgically inserted a catheter into the joint and instilled vancomycin into the joint. And these were patients that had comorbidities. Most were diabetics. Most of them were older. And they would give vancomycin into the joint once a day, and found that after a 6-week course, 17 out of 18 patients were infection-free up to 62 months later, which is really surprising. They did measure vancomycin serum levels and found that they were detectable. In fact, some of them went high, and they used that to adjust their doses. But patients, none of them had renal insufficiency. All of them tolerated the drug well. And so this made me scratch my head and think, is there a way we can treat this topically? So what I did is pretty unconventional. So I mentioned the patient had a vac dressing covering this wound. So I asked the question, can we reverse the vac pump and then instill amikacin, which the organism is susceptible to, into the wound bed a few times a day, and see if we can sterilize the wound? And that's exactly what we ended up doing. It was challenging to get approvals and to get a pump that would allow us to do it. And fortunately, the patient's father learned how to do the installation of the amikacin. We measured the patient's serum amikacin levels. They were undetectable. He tolerated this incredibly well. He left hospital with the amikacin through the pump with normal renal function, no side effects. I continued this for about a year because the wound was slowly healing. And you can imagine it's a real challenge for tissue to grow around all that metal. And once we got it to where I thought it was as healed as much as I could get it, I put him on suppressive therapy. So again, if this was a patient on an oral antibiotic, I might give them a dose a day or twice a day. But in this case, it was through the pump. So we went down to doing it daily for about a year. No Pseudomonas was detectable in the wound. I stopped it, recultured his wounds, no Pseudomonas. And today he still is left with a small wound. So this is what he looks like on an average day. And underneath that, we have a little bit of exposed hardware here, which requires just a plain dressing, and the vac dressing on this part, which is antibiotic free. So he has now been off of antibiotics for 4 years and is stable with this. Unfortunately, can't get rid of the vac dressing because this area drains an awful lot, but I'm exploring options for future dressings for him. So this was an unusual case with good success. However, multi-resistant Pseudomonas comes in many situations. So the next patient I'll talk about is one that I saw in the ICU in the summer of 2021. So I took over the ID service. It was the first day on service, and I got a call about this patient who had a multi-drug-resistant Pseudomonas in his blood. This was a patient with COVID who was on ECMO, which is extracorporeal membrane oxygenation. It's where we take the blood out of the body, oxygenate it, and put it back in because the patient's lungs aren't working well enough. And as you can see, I did not have a lot of treatment options for this patient. They already had renal insufficiency. They're hypotensive. They're very sick in the ICU. And so this was a scenario of panic to try and find a combination to treat this patient. We ended up using drugs that we didn't even have susceptibilities for. Fortunately, we have Neil Irfan on the ID service, who's a genius at helping us access things. We were able to get a little bit of imipenem relebactam and some IV phosphomycin and gave it to the patient, prayed, and fortunately he did survive this episode. But as I took over the ID service, I noticed there were an awful lot of patients with multi-resistant organisms. So I started asking myself, are resistance rates higher in these patients with COVID? So I looked back and found a bunch of patients who, 64 patients who were in the ICU with COVID. 29% of them had died and some were still on ECMO. And I looked at those patients and said, what is going on with their resistance? So in the patients without ECMO, we can see there were 12 patients, or 28% of them had a multi-resistant organism. And I've listed all the organisms there. I know this talk is about multi-resistant Pseudomonas, but you can see we had a lot of ESBLs. We had multi-resistant Klebsiella aerugines, Acinetobacter. And so it was really kind of puzzling as to, and that's more than the baseline of resistance that we normally see in our ICU. So then I looked at the patients on ECMO, and 76% of those patients had a multi-resistant organism. We had 6 multi-resistant Pseudomonas, and a bunch of ESBLs, Acinetobacter. In the patients without multi-resistant organisms, we had some of the spice organisms, and we had other Pseudomonas, which was susceptible. So really seeing a huge amount of resistance gram-negatives in this population. So then I asked the question, why do we have so much resistance in this population? Is it the immunocompromised state that they're in? Is it the COVID itself? Is it the fact that we're giving them tocilizumab and steroids? Is it that these patients all have lines? And certainly in the ECMO patients, you can't change the lines because those lines are what's keeping the patient alive. If I remove the line, I jeopardize the patient's life. Is it because we're not following a proper infection prevention and control issues? The other issue with ECMO is your volume of distribution is significantly higher because we've got them on a circuit. So we're removing blood and we're putting blood back and they're getting fluids. And so is the routine dosing of antibiotics not appropriate? And certainly if we underdose antibiotics, we promote resistance. So we actually, based on this data, did come up with a protocol to increase our dosing, particularly of beta-lactams in our patients who are on ECMO, giving them higher doses, continuous infusions, and we are in the process of setting up therapeutic drug monitoring in the lab so that we can then check drug levels in these patients to ensure that we're not underdosing. So because I have an interest in multi-resistant Pseudomonas, I decided to look at our isolates in the in our lab for HRLMP. Now, the caveat to this data is that in 2022, we switched our electronic medical record, so I only have 6 months of data. So this low part here is erroneous. But you can see over time, over the 3 years that I looked at, the multi-resistant Pseudomonas isolates have been fairly stable. I suspect that the dip here is because of COVID and people not as many patients coming to hospital. Overall, the number of patients have been stable, and the number of invasive isolates has been fairly stable. So multi-resistant Pseudomonas has been a longstanding issue for us. I think I would say that the incidence may not be significantly increasing, but the phenotype of the multi-resistant Pseudomonas is more resistant now than it was in the past. This report was published in February of this year from Public Health Ontario and the IQMH, and it looked at gram-negatives and other resistant organisms from 51 hospital-based labs, 11 private or community-based labs, and 11 public health reference labs. They submitted their data, and it looked at resistance rates for gram-negatives over time. And so here is our ciprofloxacin resistant rates for Klebsiella, E. coli, and Pseudomonas aeruginosa. And you can see the numbers wax and wane, but overall there has been an increase in resistance. They looked at, there were over 300,000 E. colis per year, 50,000 Klebsiellas, and with Pseudomonas was about 30,000 isolates per year that they looked at. And they did see an increased rate of resistance to the 3rd generation cephalosporins, going from 6.7% in 2019 to 8.1% in 2021. They saw a similar increase in resistance for blood isolates, going from 4.5% to 6.4%. And they also saw an increase in meropenem resistance. And that is the area I think that concerns me the most. Meropenem used to be our go-to drug for Pseudomonas, and it's no longer as effective, with rates in 2021 of 9% from those labs that reported. This is a demonstration of our resistance rates. Pseudomonas is the purple bar, so you can see third generation cephalosporins, about 6%, 7% for ciprofloxacin, which is a bit lower than I would have expected, and meropenem 9%. But we see a lot of E. coli resistance for third generation cephalosporins and ciprofloxacin. And the trend is in the upward range. It's not going away. The other challenge we have is access to new drugs is really limited. So this was published in 2022, and you can see lots of new drugs listed here. Sorry if it's a bit blurry, but you can see available in Canada, no, for a lot of these things. Ceftolozane tazobactam was approved in 2015. I was able to get it for my patient, and it is one that we can we can fairly regularly rely on, although there have been supply issues. And during the pandemic, when I was seeing those resistant patients in ICU, we did not have the drug available at all. Fosfomycin has been available since 2019, but it's not a very good drug for multi-resistant Pseudomonas. We are able to get imipenem relebactam through special access, and IV fosfomycin, as I mentioned, is also potentially available. There are a few other drugs that are potentially available through special access, but we have to jump through quite a few hoops to get them, and there can be delays in accessing them. So hopefully the examples I've given have shown you that as a clinician, multidrug-resistant Pseudomonas aeruginosa is very challenging to manage. And it's the bug that often keeps me awake at night for those patients where you're really not sure if you've got an active therapy on board. We've had some new drugs come available to us, but they're limited numbers. And I do think maybe it's time to explore ways to use old drugs. So I showed you a case of topical therapy. Would I do that routinely? No, but it worked in that case, fortunately, and that patient is still alive today and doing well. Or the other concern I have are we using the drugs properly? Should we be using more continuous infusions or different dosing intervals to try to reduce resistance? And with that, I will stop sharing and hand over to Brent.
Okay, thanks, Cheryl. So today I'll tell you about some relatively recent work we've been doing in the Brown Lab on these synthetic siderophore antibiotic conjugates. And so when I think about, how antibiotics are discovered, this is kind of the oversimplified process I see in my mind. So, you'll have a compound that you're interested in, and you test it in the lab on a Petri plate or in the test tube for activity. And if it kills bacteria on a Petri plate, you'll next move into your animal infection model. And if you can cure that mouse of infection, you're well on your way to you're starting your human clinical trials. And of course, I've oversimplified this, but I think if you look back at how antibiotics have been discovered in the past, this is really the process that they take. And it's been incredibly successful, generally speaking, but we're seeing fewer and fewer success stories using this process in the recent years, and especially with rising rates of resistance. I think it's worth taking a step back and reevaluating how we actually approach the antibiotic drug discovery process. And as a postdoc in the Brown Lab, I was really interested in how we can improve this first step. So actually screening for antibacterial activity. And so again, we use our lab media as our initial screening tool to find molecules with antibacterial activity. And again, it works great. So there's, most bacteria require about 300 genes to just grow and divide in standard laboratory medium. So we can consider these our in vitro essential genes. And all of our current antibiotics target these in vitro essential genes. So you can think things like involved in cell wall biosynthesis, DNA synthesis, protein biosynthesis, really great targets. But again, we haven't found any new drugs in the last few years. So one thing we know about the situation in vivo is that to actually cause disease and cause infection, not only are these 300 genes important, but there can be dozens or even hundreds of other genes bacteria require to actually cause an infection in vivo. So these are just as good a target as these 300, but we really have no great method or no great screening tool to find compounds that target these in vivo essential genes. And so one thing you can imagine, and in the very distant past, people did this, you could screen your compounds directly in animal infection models. Of course, that's today unethical and would be prohibitively expensive. So the idea I had when I started in the Brown lab was, can we find a better mimic of the in vivo environment? And this is where I thought human serum might be a more physiologically relevant media. And there's decades of work showing that some of these in vivo essential genes that we know are required to cause infection, many of them turn out to also be required to grow in human blood serum. So really, my overall project was to see if we can use a growth medium like human serum to find new antibiotics. And the approach I took was to use some high throughput compound screening to test for activity against bacteria grown in serum. So this is an experiment where we've taken 1,600 previously approved drugs and tested them for activity against Klebsiella pneumoniae grown in human serum. And so in this figure, each of these dots represents one of those 1,600 compounds, and I've highlighted in blue, these are the compounds that are killing Klebsiella in serum. And so just as a couple examples, we find antibiotics like colistin and azithromycin work much better in serum than they do in our conventional lab medium. So their MICs are greatly decreased. And this has been known for quite a while for these two antibiotics. So we then, after we were satisfied that our screening method was working, we screened another 170,000 small molecules from McMaster's compound collections. And if you're interested in this story, we published this a few years ago, but what I'll talk about now is kind of an unexpected result from these assays. So as I mentioned, this screen, we found the inhibitory molecules that I highlighted in blue. So I spent most of my time working on these. But then we realized a little bit later that there were these molecules that I've highlighted in red here. And if these ones are killing Klebsiella, these compounds are doing nothing. Presumably these ones are actually making Klebsiella grow better or enhancing growth of Klebsiella in human serum. And again, we kind of ignored these at first, but when I went and started to look at the structures of these compounds, many of them had structural similarities. So I've just highlighted in red, these compounds all have this catechol group here. And indeed, when I did some follow-up experiments, if you take dopamine and add it to blood serum where Klebsiella are growing, you would increase growth in serum, but not in conventional media. So it seemed to be a real phenotype we were finding here. And what you might recognize is that catechol group is very similar to the catechol group we find on a bacterial siderophore called Enterobactin. So gram-negative bacteria produce these iron-binding siderophores or iron-binding chelators that they use to acquire iron from the environment. So we started to think of these family of compounds that were enhancing growth as these sort of synthetic siderophores. And the reason we became very interested in these is that there was a drug approved in 2019 called cefiderocol. And this is a cephalosporin antibiotic, where they've conjugated a catechol group, similar to what we see with Enterobacter. And so the mechanism of cephalosporin and how it works, it uses this so-called Trojan Horse antibiotic approach, and I'll just explain briefly what that means. So in vivo, levels of free iron are very low. So we produce proteins like transferrin that bind up free iron in the bloodstream. So bacteria have these, like I mentioned, biosynthetic genes that produce these siderophores, such as Enterobacter, which they can then secrete to the extracellular environment. Enterobacter can acquire that iron from transferrin and then take that siderophore back up through the cell and acquire iron. And so these synthetic siderophores, the way they work, they also can bind iron, and they can take that from transferrin, and then they hijack these import systems that Enterobacter or other siderophores use, and can get inside the cell. And so the way cephalosporin works is, it's just an antibiotic that's been attached to the siderophore portion, and so this allows the drug itself, so the cephalosporin, to get in through these iron transport pathways. So it kind of tricks or hijacks the bacterium into taking up the drug, and so this is the reason for the Trojan Horse naming of these class of compounds. So, as I mentioned, we screened, about 170,000 compounds from our small molecule collections, and we find, about 100 or so of these growth enhancers, or these synthetic siderophores that enhance growth in serum. And so we thought, can we take some of these synthetic siderophores and start attaching them to antibiotics, similar to what was done for cephalosporin. And so we initiated a collaboration with the Mogollon lab, and the compounds I'll talk about today were all synthesized by Nikki Ritchie in the lab. I'm not going to show you the structures today, but the compound I'll talk about is MLEB22043, or 043 for short, and it's a conjugate of the monobactam aztreonam that I've shown here, and then we just stick the synthetic siderophore onto the monobactam. So the first experiments we wanted to do was to see, is this compound actually behaving as a Trojan horse antibiotic, like cephalosporin. And so this is the experiment we did. We're using Pseudomonas here, and the MICs in microgram per mil here. The MIC for aztreonam is 8 microgram per mil in conventional. This is our conventional testing media, and for 043 is 1, so it works a little better than aztreonam. If we test this in iron depleted MHB, so this is the MHB where we removed all the iron, and this is the clinical media used for testing for cephalosporin activity. But you can see we get a 16-fold reduction in MICs for our conjugate, and not much of a change in the MIC for aztreonam. If we add back iron to that iron depleted media, now we see that sharp increase in MIC for our conjugate, and so this indicates to us that this is an iron dependent Trojan horse compound. And again, if you did the same experiment with cephalosporin, you would see very similar results. So we were happy that conjugation of our synthetic siderophore to aztreonam was kind of transforming it into this Trojan horse monobactam. Because of the pretty good activity I saw against Pseudomonas, I then went and determined MICs against 97 clinical strains of Pseudomonas. So the Wright and MacArthur labs at McMaster have put together a clinical collection of drug-resistant gram-negative pathogens, and so that's where these 97 clinical strains came from. And we measure this as what's called an MIC90, so the concentration of drug required to inhibit 90% of those clinical strains. So for aztreonam, that concentration is greater than 16 microgram per mil, so higher than the highest concentration we tested in the experiment. And for the conjugate, our MIC90 is 0.5 microgram per mil. So quite potent compared to aztreonam against this clinical collection. And just for reference, the breakpoint for Pseudomonas is less than or equal to 8 microgram per mil for aztreonam. So presumably we're at a good, we have good activity that might be relevant moving forward. Unfortunately, when we start looking at other bacteria, some non-Pseudomonas bacteria, the results are not as promising. So a couple strains of Klebsiella here, and E. coli and Acinetobacter, and aztreonam and O43, you can see both are relatively ineffective. We have a strain of Klebsiella that actually lacks the carbapenemase. So this is a beta-lactamase gene KPC3. And interestingly, when we test that strain, now we see activity for aztreonam and our conjugate. So this led us to believe that the lack of activity we're seeing here for our conjugate is likely beta-lactamase mediated. So we wanted to investigate this a little bit further. And just a very brief overview or primer on beta-lactamase nomenclature. We have 4 classes of beta-lactamases, class A, B, C, and D. Class A, C, and D are our serine beta-lactamases, and then class B are the metallo-beta-lactamases. We'll often hear about NDM1, being a pretty scary enzyme because it hydrolyzes most beta-lactams. But interestingly, it doesn't work against monobactam. So this will become important here in a few slides. So just keep that in mind. So we wanted to know what's the susceptibility profile of our conjugate to different beta-lactamases. And so the right lab has their antibiotic resistance platform that is a bunch of E. coli strains that express single beta-lactamases. So we tested aztreonam, our conjugate, and then we've included ceftazidime and cefiderocol as comparators in this experiment. So these are the MICs for an E. coli strain that has no beta-lactamase. If we express the metallo-beta-lactamases, you can see aztreonam and O43, there's no change in MIC. So as I mentioned, these are not affected by metallo-beta-lactamases. Ceftazidime, the MIC goes above 2 microgram per mil, and then we see less of an effect against cefiderocol because of its known stability to beta-lactamases. Then the story with serine beta-lactamases is the opposite. So aztreonam and O43 are both affected by this selection of serine beta-lactamases. This is a known phenotype with aztreonam. It's again, susceptible to these serine beta-lactamases. But what's nice is that we now have really good inhibitors of these serine beta-lactamases. So avibactam is a broad spectrum serine beta-lactamase inhibitor that works against class A, C, and most D beta-lactamases. And because our monobactam works against class B metallo-beta-lactamases, in theory, we should have really broad spectrum if we combine our monobactams with avibactam. So we can go back to these E. coli strains that are expressing our beta-lactamases and do that exact experiment. So we take aztreonam and combine it with avibactam, and now you can see the MICs drop substantially against these serine beta-lactamases. And then the same experiment, but with our conjugate. And again, now our conjugate works against all of these beta-lactamase expressing strains. So with this in mind, we now know that our conjugate is still susceptible to these beta-lactamases, or the serine beta-lactamases, and we can add avibactam to overcome that resistance. We went back to those few gram-negative pathogens that I mentioned, so the Acinetobacter, E. coli, and Klebsiella, and we can now test them in the presence of ace-trionam and avibactam, and we get now a drop in MIC to susceptible levels for E. coli and Klebsiella. Interestingly, no change in MIC against Acinetobacter. Whereas with our conjugate, now we see a drop in MIC for all strains tested, including Acinetobacter. So adding avibactam is obviously overcoming some of these strains must be producing some serine beta-lactamases that avibactam is able to overcome. What I thought was pretty interesting is this result here with Acinetobacter. So even adding avibactam did not have any effect on ace-trionam's MIC, but we did see a decrease in MIC for our conjugate. So I tested, a total of about 10 strains of Acinetobacter, and you can see that ace-trionam, either alone or in combination with avibactam, has no real benefit. All of these strains are more or less resistant. Our conjugate, actually as a monotherapy, works really well against most strains of Acinetobacter. And in the case of this one resistant strain, adding in avibactam now gives us coverage against that strain. So it's clearly expressing some beta-lactamase that requires the addition of avibactam. So we've seen activity against some Klebsiella, E. coli, Acinetobacter, and Pseudomonas. What I was really interested in testing was, or at least comparing, how our conjugate fared against other beta-lactams. So kind of putting it through a stress test, saying, if we take some of the most resistant strains we can find, how does it stack up against some of the existing antibiotics out there? So I took about 10 strains of Pseudomonas that were resistant to meropenem, and we have because these are all genome sequenced, the MacArthur lab has the predicted beta-lactamases for each of these strains. And these are all meropenem-resistant strains. They're variably resistant to ceftazidime or ceftazidime avibactam. They're generally all resistant to ace-trionam or the combination of ace-trionam and avibactam. And I will note, Cas-Avi is an FDA-approved combination. As-trionam-Avi is still in clinical trials. I think maybe phase 2 or 3. So it's not an approved therapy yet, but in any case, it has no use, it seems, against Pseudomonas. So cefiderocol, again, as a comparator, is a really great drug. It works against all these strains of Pseudomonas. Our conjugate, as a monotherapy, again, works against all these strains, except for this one case. And in this case, if we add back in avibactam, now we get all strains susceptible to the combination of our conjugate and avibactam. So again, just a handful of clinical strains. At least compared to cefiderocol, it seems like this combination is holding up pretty well. And then finally, again, looking to find kind of the most resistant strains we can get our hands on. These are 2 strains of Klebsiella expressed or predicted to encode a variety of beta-lactamases, including both of these have the NDM1 enzyme. And so they're very resistant to basically all antibiotics, meropenem, ceftazidime, Cas-Avi, ace-trionam. The combination of ace-trionam, avibactam now works against these strains. Cefiderocol works really well. Our conjugate as a monotherapy, not very effective, but if we add in avibactam, we see the MIC is quite low. So with that, I'll just summarize kind of the story here. The conjugate, adding our synthetic siderophore to this monobactam, improves our activity against Pseudomonas and Acinetobacter. So even as a monotherapy, we get activity against most strains. By combining it with avibactam, there's really, at least in our collections, we haven't found a strain that it doesn't, a gram-negative strain it doesn't work against. So it seems to be the real stress test will be starting to compare it against Ace-Trionam, avibactam resistant strains and cefiderocol resistant strains, and so we're hoping to test that soon. And then more broadly, I think this work shows that, I mentioned the serum screening assay where all these compounds were discovered. I think it shows, it has more, it's broadly applicable to finding both new antibiotics as well as these synthetic siderophores that we're excited about attaching to other antibiotics. So this has been a collaboration between our lab, the Mogollon lab, and the Ardal group in Norway. And Haley has sent me a lot of strains for testing these compounds. And then, as Laurie mentioned, I've been hosted by Tracy Ravio here in Edmonton for the last couple of years to continue on this work. And that's it.
Thank you both so much. We have some questions posted. So the first question is for Brent. Just wondering if uptake of your compound is dependent on TON-B, and if you think mutational resistance of siderophore uptake mechanisms might be a concern in vivo with Pseudomonas.
So yes and yes to both. TON-B, we have at least in E. coli. So we're working with Laurie's lab on this now as well. And but I've done the experiment with E. coli, and a TON-B mutant is resistant to, or at least we see a substantial increase in MIC to this compound. So it is using the siderophore uptake pathways. And then the second part was just if we were concerned in vivo.
Yep.
Yeah, I think that's certainly a concern. With cefiderocol, I feel like the mechanisms of resistance that you can get on a Petri plate seem to be a little bit different than maybe that you'll see in a patient. It's really easy to get a mutant on a Petri plate. It seems like you need a combination of some beta-lactamases plus mutations to get resistance in a patient. You might not expect to see a TON-B mutant arise in the clinical setting, whereas you can get that mutation very easily in the lab. So I think we'll see in the coming years as cefiderocol is used more and more.
The other question is more of a comment. Amazing work. Excuse me, when are you going to publish it?
Well, I don't know. We'll see. There's, this is all in vitro so far. I think that the big test is going to be, these things might be terribly toxic or something. So it's, I think we're at the very beginning stages, and there's a lot more MICs to do still, and then eventually get it in vivo.
Cheryl, have you had much experience with cefiderocol or people using it?
We can get it. I know we used it for one patient. I've not used it. I think it's quite a few hoops you have to jump through. We actually had to get the patient to sign off on some of the black box warning stuff with it. So I know it was a bit of a challenge, but no, I've not used it.
Yeah, I was just thinking about, being able to learn about the pattern of resistance requires that someone actually tries to use it in the first place. Luke has a question for Brent. He wants to know if you've tested clinical isolates for heteroresistance to your compound.
No, but so cefiderocol displays this phenomenon of heteroresistance.
Maybe you could just explain for people who don't know what that is.
Yeah, I honestly don't exactly understand it, but basically, if you plate a population of bacteria on agar plates containing cefiderocol, you get a very small subpopulation can show resistance to cefiderocol, and one of the mechanisms is through gene amplification. So they amplify the number of beta-lactamase genes, which amplifies expression. So interesting with heteroresistance, if you do that same experiment and you take cefiderocol and you add avibactam, you don't see heteroresistance anymore. So it seems like if you add this inhibitor of the beta-lactamases, you no longer get heteroresistance. So I think that's will be important going forward, exploring maybe some combination therapies, even with cefiderocol, because it seems to be a concern in maybe clinically. I think people have this concern for heteroresistance.
Thanks. And Cheryl, I have a question for you just about, you were talking about COVID patients and ECMO. I know a lot of COVID patients were just ventilated.
Yeah.
Did you see a lot of Pseudomonas and Klebsiella in those ventilated patients as well?
We saw some, so that would be in the group that was that 27% versus
Right.
ECMO patients, where it was in the 70s. So we did see some. I think it was probably a little higher than we see it routinely in the ICU, but we see multi-resistant pseudo in the chronically vented patients in our ICU anyway. And we wondered if it was related to the dexamethasone that they were getting, but yeah.
Do you have any feel for whether it's like you're having any clonal spread of these organisms? Like, do you guys do any genotyping to figure out if you're having some kind of problematic strain?
Yeah, so we will do typing if infection control thinks that there's an outbreak. And I don't believe there was any during that time. They were sort of sporadic cases. And again, the resistance wasn't just Pseudomonas, right? It was all multi-resistant organisms, right? We had ESBLs and carbapenemase producers, and it was a mess up there.
Just what you need on top of COVID, right?
Yeah.
Okay, there's a few more questions here. So from Dr. Coombs for Brent, can the conjugate enter bacterial cells through stealth siderophores like IRO-N or others?
We don't know yet. So that's one of our major goals in the next few months is to really define the uptake pathways. And like I said, we're working with Lori's lab and Derek's done some experiments to start to unravel the repertoire of iron uptake pathways these conjugates can use. So hopefully in the next few months we'll have a complete story on the uptake pathways.
Yep, stay tuned. A question for Cheryl from Melissa. Are there any cases of phage therapy you've come across in your work in a clinical setting, specifically treating MDR Pseudomonas?
No, there aren't. And in fact, with the patient that I presented, phage therapy was one of the avenues that I was exploring. I couldn't find, I found there was some place in Russia that said that I could get phages from them, but it seemed a bit sketchy, didn't actually pan out. So we ended up going with the topical therapy. But I did tell Lori before this presentation that if he runs into problems again, I'm going to need a phage because I don't have anything else for him. So
yeah, it's pretty, I'm sure that for those patients, it's very scary to know that they're like, one drug away from no solution too. And, I really felt for your patient when you said that he just wanted to give up because of all the side effects of the drugs, right? Like
yeah,
you don't,
the general consensus, some of my peers said to me, he's going to die because we just didn't seem to have anywhere to go. I'm really grateful that my topical therapy worked. And I've had lots of people ask me since, should we use topical therapy? I don't think it's a first line therapy, but certainly in this patient, it was a good option. And now 4 years later, no antibiotics. So
yeah, I know there's a lot of Hail Mary type of, putting solid drug on prosthetic infections just because you don't really have many other option type things. Okay, there is another question from Brody for Brent. He says, really, really interesting. One limitation of aztreonam is lack of coverage of gram-positives, for example, polymicrobial infections. Have you done any screening adding siderophores to other beta-lactams with gram-positive coverage, such as meropenem?
No, that's a good idea. So yeah, aztreonam, this conjugate, actually, I haven't tested against any gram-positives, but presumably it won't work. But really, our ultimate goal is to take as many of these siderophores we've found attach them to as many antibiotics as possible, and kind of do those sorts of experiments where. But I will say that the chemistry of all this is very challenging. So it's not just one day in the lab that to make these conjugates. It's a very challenging process to make some of these conjugates. So we'd like to attach as many siderophores to as many antibiotics as possible, but it's just sometimes it's not so straightforward.
Yeah, and I think getting them into the paraplasm of a gram-negative is one thing, using these nutrient transporters, but starting to tag them onto things that have cytoplasmic targets is a whole other ballgame, because then you also have to get through the plasma membrane.
Oh yeah, and nobody's been able to really make that work, despite 40 years of people trying this. Getting a cytoplasmic drug in has been pretty unsuccessful.
Well, I was really surprised to learn that how large compounds cross the cytoplasmic membrane is really not very well understood. People just kind of shrug when you ask about this, right? Because we've been looking into this with some of the very large natural product antibiotics, and it's not very clear how they managed to do that. So. Okay, I don't see any more questions. So thank you both again for great talks on my favorite organism. And thanks to the audience for joining us this morning. We'll see you next month.